system of cellular immunity, T-lymphocytes, Department of Internal Disease. My name is Izmailovich Marina Rashidovna, Assistant Professor. Cellular immunity, this type of immunity that includes specific and non-specific immune response. Non-specific cellular immunity or innate immunity is essential for providing immediate protection against the infections and preventing the spread of pathogens while the adaptive immune response is being activated. And the function of non-specific immune response perform the cells as neutrophils, eosinophils, basophils, monocytes, macrophage, dendritic cells, and natural killer cells. And other one type, adaptive immune response or specific immune response, is crucial for targeting intracellular pathogens such as viruses and some bacteria and plays a significant role in tumor surveillance. The function of adaptive or specific immune response performed T-lymphocytes. Here you can see all cells that include cellular immunity. This include granulocytes or agranulocyte cells. And we talk about the T-lymphocytes. T cells or T-lymphocytes are small lymphocytes, typically measuring about 9–10 microns in diameter. They possess a large nucleus with dense heterochromatin surrounded by a thin cytoplasmic body containing few organelles like mitochondria and ribosomes. When activated, T cells enlarged and increased their cytoplasmic volume and organelle content. T-lymphocytes have a surface markers. It's T cell receptor responsible for recognizing specific antigen presented by major histocompatibility complex on their cells. CD3 marker found on all T cells. CD4 found on T helper cells only. CD8 marker found on cytoplasmic T cells. CD25 and 69, its activation markers. FOXP3, its regulatory markers. CCR5 and CXCR4, its chemokine receptors. And CD44 and CD62, its memory markers. T cell receptor is a critical protein complex found on the surface of the T lymphocytes. The T cell receptor is primarily composed of two different protein chains. Here you can see it's alpha and beta chain. The alpha chain, one of the two chains that makes up the T cell receptor responsible for recognizing specific antigens. And the beta chain, the second chain that pairs with the alpha chain, also involved in antigen recognition. And there is two domains, it's variable and the constant. Variable domain, each chain has variable domain that form the antigen binding site. And these regions are crucial for the specificity of T cell receptor, allowing it to recognize a diverse array of antigens. And the constant region provides structural support but do not participate in antigen recognition. And now I'll talk about the differentiation of T lymphocyte. There is T cells originate for hematopoietic stem cells in the bone marrow, and these progenitor cells migrate to the thymus for the maturation and differentiation. It means that T lymphocytes differentiation has the two stages. The first stage is antigen independent, occurs in the thymus and does not depend on the introduction of the foreign agents into the body. And the second stage is antigen-dependent differentiation, occurs in secondary lymphoid organs, and is triggered only when the antigen enters the body. The antigen-independent differentiation in the thymus is accompanied by a change in the surface markers of T lymphocytes. It includes the following stages: rearrangement of T cell receptor genes and formation of a full-fledged receptor. The next, the formation of major subpopulations of CD4+ and CD8+ T lymphocytes. And the last one, immigration of mature T lymphocytes from the thymus. The main markers of differentiation of T lymphocytes in the thymus are characteristic co-receptor molecules. It's CD4 for T helper cells, CD8 for cytotoxic T lymphocytes, and T cell receptor for all types of T lymphocytes. In thymus occurs the selection of T lymphocytes, the positive and negative selection. And it is very important because the positive selection, the process by which T cells that can successfully bind to major histocompatibility molecules, which at least a weak affinity are selected for survival. And this process occurs primarily in the thymic cortex. The thymocytes that do not bind effectively to major histocompatibility complex molecules undergo apoptosis. And this ensures that only functional T cells capable of recognizing MHC complex survive. And positive selection is crucial for establishing and functional T cells repertoire capable of recognizing foreign antigen presented by MHC molecules, while ensuring that T cells can interact with self-MHC, which is essential for their activation and peripheral tissues. And the other one, negative selection. This process eliminates T cells that bind too strongly to self-antigens presenting on major histocompatibility complex molecules, and thus preventing autoimmunity. And this process occurs mainly in the thymic medulla. Thymocytes that exhibit a low or moderate affinity for self-antigens are allowed to survive and exit the thymus as major self-tolerant T cells. Negative selection is essential for maintaining self-tolerance and preventing autoimmune diseases. It ensures the mature T cells do not react against the body's own tissues and those safeguarding against harmful immune response. Antigen-dependent differentiation. When T cells leave the thymus, they migrate continuously to the secondary lymphoid organs to be primed and differentiate into effector cells after meet with a foreign antigen. And foreign antigen is presented to T lymphocytes in the form of peptide by a major histocompatibility complex molecule class 1 or class 2. And CD4 cells or T helper cells recognize foreign antigen in complex with the class 2 MHC. During the development of the immune response, they become T helpers that produce cytokines necessarily for the destruction of cells damaged by the pathogens. And the other CD8 cells interact with the foreign antigens in complex with the MHC class 1 molecules. And they perform the function of cytotoxic T lymphocytes. They directly kill the cells on whose membranes they have recognized epithelium. We have several types of T cells. T cells include effector T cells that perform function of cytotoxic T cells, helper T cells, and regulatory T cells. And memory T cells have a longer lifespan as their name found empty. Start from the T helper cells. T helpers are represented by three subpopulations: T helper 1 type, T helper 2, and regulatory. T helper 1 is primarily involved in responses against intracellular pathogens and produce interferon gamma. T helper type 2 focus on combating extracellular parasites and allergens and produce interleukin 4, interleukin 5, and interleukin 13. And the last one, regulatory B cells, help maintaining immune tolerance and prevent autoimmune diseases by producing anti-inflammatory cytokines like interleukin 10. Cytotoxic T lymphocytes recognize infected or abnormal cells through the T cells receptors and through two mechanisms. The first is release the cytotoxic granules containing perforin and granzymes. Perforin form pores in the target cell's membrane and allowing granzymes to enter and induce apoptosis. And the second one, the cytotoxic T cells, it has a fast ligand interaction which bind to fast receptors on target cells, triggering apoptosis via a different signaling pathway. Memory T cells include central and effector cells. Central memory T cells reside in lymphoid tissues and have the ability to proliferate extensively upon re-exposure to their specific antigens. But the effector memory cells circulate in peripheral tissues and can quickly exert effector functions such as cytokine production or cytotoxic activity upon encountering their specific antigens. And memory T cells can be generated directly from naive T cells or from activated effector T cells. They can survive for years or even decades. And memory T cells are significant in transplantation medicine, where they can contribute to graft rejection, vaccine development, and immunotherapy. Thank you for your attention.